Hello class, welcome to the third video of chapter 5. And in this video, you will learn about the intracellular and extracellular enzyme. What is intra? What is extra? So here, intracellular enzyme, the meaning is the enzyme that are synthesized in the cell and used in the cell. Synthesis in the cell and used in the cell. That means synthesized in cell A and used in cell A. Means intracellular enzyme. And what about extracellular enzyme? Extracellular enzyme means synthesis in cell A and used in cell B. I repeat, synthesis in cell A but used in cell B. So you look at here, enzymes that are secreted outside the cell. I repeat, extracellular enzyme means this enzyme synthesis in cell A but used in cell B. Okay, what is the example of intracellular enzyme? Here, there is hexokinase enzyme. Is the example of intracellular enzyme. And what is the example of extracellular enzyme? Yeah, trypsin enzyme. Trypsin enzyme, you may look at here, produced in pancreatic cell but used in duodenum cell. That means synthesis in cell A but used in cell B. So this slide settled. We look at the next slide. Next slide is talking about the production of extracellular enzyme. Again, what is the meaning of extracellular enzyme? Means the enzyme synthesis in cell A but used in another cell. Here, I minimize the PowerPoint when I explain this slide and I show you this picture. This diagram is the production of extracellular enzyme. Production of extracellular enzyme. So the first one, what is enzyme? Actually, enzyme is a protein. It's a type of protein. So protein synthesis in which part? Yes, ribosome. So the first one, the first station start from ribosome. Ribosome is the site of protein synthesis, so ribosome here. The ribosome attached to RER. After the proteins are synthesized by ribosome, the proteins will enter the lumen of RER. Enter the lumen of RER. Lumen means lubang of the RER and are transport through it. 所以它就一直走了那个 protein transport throughout the lumen of RER， 然后一直走 yes， 走到去外面 outside the RER， 它会做什么？ Once the protein reach the end here lah, the end of RER, the membrane bursts off to form transport vesicle. So the protein will leave the RER by using transport vesicle. That means protein 它会让它自己住在 transport vesicle 里面。Transport vesicle actually like a bus leave from RER， 从 RER 那边出来的 bus 那就是 transport vesicle。然后这个 transport vesicle 它载着 protein and this transport vesicle 它会 move to where? Yes, the transport vesicle contains protein will move towards Golgi apparatus. Transport vesicle which contain protein will move towards the Golgi apparatus. 为什么 Why the transport vesicle contain protein move towards the Golgi apparatus and combine or fuse with the Golgi apparatus? Why? Because Golgi apparatus is a factory， 是一间工厂。然后 Golgi apparatus this factory will modify 改装 will modify the protein into enzyme。所以就是因为有 Golgi apparatus， then the protein the typical protein will modify into enzyme and secreted in secretory vesicle。 所以 after the modification of protein into the enzyme， 现在这个 enzyme 它要出来了，要从这一个 Golgi apparatus 出来了。怎样出来？通过 secretory vesicle。 secretory vesicle 就也是一种 bus 来的。That's a form from the tip of the Golgi apparatus. So the secretory vesicle 它里面就有 enzyme 咯。然后这个 secretory vesicle which contain enzyme will move towards the plasma membrane of the cell and fills with the plasma membrane to secrete the extracellular enzyme out of the cell。 所以在这边 this is a cell A， 这个是一个 cell A， 然后 this one is the place or is the space outside the cell A。 那么现在 extracellular enzyme 它会 extracellular enzyme is secreted out of the cell。 那么现在 extracellular enzyme 它就 move outside of the cell， move out of the cell。 完成。 Now we look at the next question. Try to answer this question. The following are organelles in a pancreatic cell. Organelle. Which of the following is the correct sequence of the organelles in the production of lipase enzyme? That means lipase enzyme is an example of what? Yes, it's an example of extracellular enzyme. I repeat, lipase enzyme is an example of extracellular enzyme. So, how to produce an extracellular enzyme? Refer back to this diagram. So the first station is what? Yes, ribosome. The first station is ribosome. And the next station, the second station. If refer to the choices here, the second station is, yes, transport vesicle. From ribosome to the next station is transport vesicle. And the third station is Golgi apparatus. And the next station is secretory vesicle. So what is your answer? Yes, the answer here is B. From ribosome to transport vesicle, and next to Golgi apparatus, and the next is secretory vesicle. So this question settled. And now we try to answer another question. Which sequence is correct in the production of the extracellular enzyme? So from the first station, yes, is Z, the first station. And after that, to Y. And after that, to X. Settle. So the answer is Z, Y, X. Here, what is Z? Ribosome or RER. And what is Y? Golgi apparatus. And what is X? Yes, the secretory vesicle fills with the plasma membrane of the cell and secrete the extracellular enzyme out of the cell. 那么 this question and this slide is the last slide for this video. Bye.